Dr. Akshay Srivastav is an associate professor at National Institute of Pharmaceutical Education and Research, Ahmedabad. He did his PhD in bioengineering from IIT Kanpur and his postdoctoral from National University of Ireland. He was also a DBT Ramalingaswamy Re-Entry Fellow. He is currently working in the area of biomaterial design and fabrication, tissue engineering, in vitro models, bioseparation, and biosensors, finding novel therapeutic strategies for tissue regeneration and developing in vitro platforms to understand disease pathology. In today's lecture, Dr. Akshay Srivastav will be talking about the recovery of spinal cord injury, also called as SPI. The major medical problem associated with SPI is neuroinflammation and neuronal degeneration at the epicenter, due to which the patient has a high mortality risk in the first year after injury. We'll be talking about the tissue engineering based strategy, which uses highly porous, mechanically spongy, and electroconductive hydrogel material to suppress neuroinflammation and promotes faster spinal regeneration. Without further ado, I would request Dr. Akshay to present his talk. Over to you, sir. Thank you.
Okay, thank you. Thank you for the introduction, Jetal, and I must thank Dr. Banod for the invitation and giving me a chance to talk about my work in this I think very interesting workshop. And I've seen the all the speakers, the experts in their field, and they were talking about. Probably they have all talked about the methods that they have adopted to do the high-end research, and that's what it means in the workshop. Probably you all get aware about the new technologies and how those experiments were conducted. And in today's talk also, I have tried to not only showing the result, but probably will try to tell you that how those experiments were planned, because that probably will help most of the students and out there. So I'm going to share my screen. Give me a moment. Yeah, I hope it is visible. I hope it is.
Yes, sir.
Yeah, I'm keeping it full screen. Okay. All right. So uh, uh, in today's talk, I will be talking about the electroconductive and mechanically spongy material which our group has developed. And it the material itself was developed just because we wanted to enter into the space of spinal cord repair and regeneration. And if you look at why we have chosen this area, and spinal cord injury is a very serious concern worldwide, and it also has a very high socioeconomic burden that significantly affects patients. And if you look at that, how one can get the spinal cord injury is the large proportion of people are getting injured during the vehicular accidents, and followed by falling from the height, sports, and during medical surgeries also there are some reports when the spinal cord injury is happening. So altogether, whenever this is it's such a delicate tissue in the body that just a mere touching of it, and just a some shock, or the major injuries, it actually causes the paralysis, and that is completely irreversible loss of the locomotion ability. So if I only talk about India, around 15,000 cases per year, it is per year has been seen in last few decades. And this is a kind of an average of it. It looks like a small number, but impact is really high, because it further burdens the family, and even the hospitals and everywhere. And when we try to look at what is the problem with this the repair process, and we found that there are multiple reasons, and one of the most prominent reasons are the neuroinflammation, and thus very limited axonal regeneration capability. So we further looked into the spinal cord injury pathology, which is a multifaceted, and it attenuate the inflammation, promote axonal axonal regeneration at the epicenter is the prime requirement. If we can control the inflammation, if we can promote the axonal regeneration, then probably we will be able to achieve the faster recovery. And if you see, if you leave these after the injury, if you leave the spinal cord, injured spinal cord as it is, it will actually uh, develop a fibrosis like structure, and they will have a lot of microglia activity, some inflammatory cytokines, and some astrocyte become reactive, and it never it further try to repair, which is the body's own mechanism to do that. Okay, but while doing that, they don't do it correctly. They undergo scar formation and it never repair, and probably most of you know that the nerve cells are very having a very poor regenerative capacity. So altogether, from various literature, what we got to know that the neuroinflammation is one thing which we need to tackle. The another thing is they have very poor intrinsic neuronal regeneration ability. Okay, so there are plethora of you know approaches going on, and it has been seen that the most common approach what even clinician adopt is the pharmacological. They give the medicine, and they try to relieve the pain, they try to stimulate the motor neurons, but for that the multiple dose is required, and I mean, I have written limited axonal regeneration, but in most probably there is no axonal regeneration by pharmacological approaches, what we know so far. Okay, then another area is of cell therapy. It's very famous, like stem cell. You give stem cells, you try to regenerate everything, but why none of these stem cells technologies and therapies are not so successful? The main reason is the being because they go in an environment which is highly inflammatory in nature. In such an inflammatory nature, the stem cells either differentiate into some different lineages, or they get undergo apoptosis and they get die. Okay, so, and there are some, of course, the source of stem cell is still under question, and there are ethical reasons behind that. It's a highly time consuming work. So there are some other physiotherapy like approaches are also available, where the spinal stimulation was done some because of giving some electric current. It has shown some potential, but still it was not providing the 100% success in repair process, particularly in regeneration of the spinal cord. And there's a plethora of biomaterials have been fabricated, and they are tested for promoting axonal regeneration and controlling or decreasing the neuroinflammation. Also, you will see so many papers are talking about that. Okay, and these therapies, which include biomaterials, they try to fill the cavity at the injured site, try to deliver the drug, try to deliver the stem cells. So this is what the various approaches are going on at the moment. And however, the main barrier for axonal regeneration during SCI is actually their inability of neurons to regenerate, and again I will reiterate that neuroinflammation. And in the neuroinflammation, I'll particularly put emphasis on the astrocytes and the microglia. They play a very important role in promoting neuroinflammation after spinal cord injury. Okay, and the formation of glial scar near the injury site is again hinder the regeneration process, and astrocytes respond to the injury, and they become reactive. That is called reactive astrogliosis. And the glial scar, they consist of the astrocytes, microglia, macrophages, and some ECM components. And altogether, they make such a tough matrix that it just fill the gap. It does not allow the regeneration process altogether. Okay, and the macrophages and astrocytes, they depend on the signals. They can actually either can become reactive, or we can say pro-inflammatory, or in certain signals they can become anti-inflammatory. And if we can make them anti-inflammatory, probably we will be able to accelerate the neuronal regeneration. And that's what overall thought came out from our literature study. And one time we have also visited in Ahmedabad, the local hospital, where there is a professor called Aruna Waninker. She's trying to, repair the spinal cord by using stem cells, and we have discussed it in greater length. And we definitely identify the clinical gap also in this perspective. And whatever treatment we have so far, we know is like the average axonal regeneration time what they offer is greater than 8 weeks. Okay, both in the preclinical and clinical studies, and even after 8 weeks, the recovery is quite partial. So with this, we have identified the problem, and with that, we started designing the material. So there are many things which are important, and we know that there are some physiotherapy approaches are there, and we know spinal cord and nerve cells love electric conduction, so we try to involve electroconductive in the nature in our material. And similarly, if you talk about the unique properties which probably may accelerate the axonal part of the spinal cord, then you need a conduit like structure which is having a porous network, so that at the broken ends the cells can approach each other and try to connect and form a bond and then form an entire tissue. So three-dimensional network, porous network, is entirely is very important for making the connections, as well as they are important to have a proper nutrient to the cells which are growing in that gap region. The another important feature which is required, that it needs to be mechanically spongy. So again, the mechanical pressure has shown in recent time that they modulate the cellular expression, they initiate the regeneration process. Many genes have been identified recently. Some of you probably know the Nobel Prize also goes into the piezo electric kind of a materials. So the mechanical force is extremely important. So we wanted to have, if we want to tune our material in such a way that it can provide the mechanically spongy material, and electric conductivity. Of course, the quite rationale is over there why we wanted to have electric conductivity as well. Uh, and with altogether material properties, what we wanted is that we should be able to suppress the neuroinflammation. And in future, probably either this material alone or along with the combination of stem cells can provide a full solution for the faster axonal regeneration. And the idea is that when the material will be implanted at the injury site, and then the material what we are using can be placed, and after placing a material, it provides all the material properties for the making a connections and helping in repairing and decreasing the inflammation. At the same time, we wanted to put electric stimulus from the outside, and we hope, and that is one of our hypotheses, that having a material implant and giving electrical conductivity from outside, we will be able to promote axonal regeneration. Probably we will be able to control the macrophages polarization, that M1 type or M2 type. M1 type is a pro-inflammatory type, and M2 type is an anti-inflammatory type. And we want that after the injury, when we implant the material, it should be able to convert macrophages more toward the M2 phenotype. But we always need like a balance of both. It should not, so but the ratio should shift more towards the anti-inflammatory type. Okay, and then we can the material allows the proliferation of cells, and and the material should be ultimately degradable in nature. And that was, with that solution, we actually develop a material. And in the material selection part, we selected collagen type 1 because that is a type of a material which is largely present in many tissues, is quite biocompatible, biodegradable, and at the same time collagen type 1 is present in large quantity in the spinal cord as well. So it's kind of a biomimicking material what we have chosen. The another choice, we had so many polymers and all, but we have chosen graphene as a material. Okay, so it is seen then in some of the recent literature that the graphene have capability to modulate the macrophages properties. Okay, it has shown that graphene is actually modulating the inflammatory responses as well. However, higher concentration of graphene is toxic as well. Okay, and that is around 2% of graphene is actually toxic for the body. So we started, incorporating graphene with our collagen material, and we have screened with the various concentration, and we actually followed the path of cryogelation. So cryogels, those who doesn't know about it, it's a simple conventional hydrogel synthesis procedure. The only thing what we do here is we synthesize the hydrogel and freezing temperature at sub-zero temperature. Okay, so we have optimized the condition of at around minus 12 degrees Celsius for 15 hours. If we keep it at minus 12 degrees Celsius for 15 hours after mixing the graphene and collagen, we get the this kind of hydrogel. And this is this, you see the blackish nature. This is means that graphene is incorporated. Now if you look at the structure of graphene, so graphene itself, if you try to mix with collagen, it will only be entrapped within the collagen material. So what we did, we first functionalized the graphene with the amino group. Okay, why we wanted to functionalize the amino group? Because we wanted to conjugate this graphene with the collagen. So collagen has both the carboxyl and the amino groups. We have chosen we have chosen the carboxyl group of the collagen and amino group of the functionalized graphene, which can be conjugated by a zero-length cross linkers called carbonamide and hydroxysuccinimide. By using this chemistry, we have made a highly spongy type of material. Okay, you can see. Okay, somehow this video is not working, but what we wanted to show here is with this forceps we can press this material more than 90%, which, and then if you again put it in water, it reswells, which is a very unique and very kind of a specific property of cryogel, which it defines that this is a cryogel like material and having a good porosity. So with this material, we further wanted to see what is the cross-linking efficiency. So we did cross-linked with the various concentration of graphene, and you can see that the maximum cross-linking what we could achieve is at zero point 0.5 weight by volume graphene. Okay, going beyond this, it's actually it's a saturation limit, and graphene does not involve in the cross-linking mechanism. And similar trend what we have seen in thermogravimetric analysis and in dynamic calorimetric assay analysis, we found that this 0.5% graphene, though because it is cross-linking more and thus showing more stability based on in the temperature. And we compared our result with a conventional collagen hydrogel, which is cross-linked with carbonamide and NHS alone without any graphene. Okay, so we can see that with a conventional collagen hydrogel, we can achieve a better cross-linking when we incorporate the amino graphene in our system. So with this, we are quite sure that material is spongy, it is stable, and it is having all unique properties in terms of stability. And first thing, now we one by one we will start proving all the properties, like graphene is electroconductive in nature, whether collagen itself is electroconductive, but by adding it is not sufficient. So by adding a graphene, we have enhanced the electric conductivity of the collagen, and which is again apparent here when we compare, like 1% graphene is showing the highest electric conductivity. The current which is passing is at different voltages is very high compared to the lower concentration of graphene, and thus we have again chosen, we are moving towards like how we have towards the 0.5% of graphene in the cryogel. Okay, so overall conductivity wise, 0.5 shows the significant difference between 1% and 3.1%. And with that, we moved ahead and did the porosity analysis. With this data, what you can understand, the overall message is that swelling index, the pore volume, and the interconnected word volume, you see more or less these all materials are fairly same. It means EDC NHS crosslink collagen alone, whatever porosity is there, we are actually having the same. Okay, so in terms of the volume which is present in the material, and there some images also looks fairly same. The porosity ranges are quite different though, but overall volume which is hollow and which is like having a porous network, is quite similar. We further wanted to know what actually unique about how graphene is going to influence the porosity. And again, this is something we found little different in microcity analysis, where we found that when you cross-link with EDC, you see more fibrous-like nature is there in the material. Okay, though the overall volume, free volume is same, but nature is more fibrous in nature. But when you cross-link with 0.1% and 0.5% graphene, in the collagen, then you see this fibrous nature is totally diminished, and more, you see some white patches like colonies kind of structure, which is like a thick pore structure, and also it is because of the aggregation of the graphene in the whole system, which probably will be useful for a system. So, but overall porosity does not change. We further looked the pore sizes, and the pore size of 0.5% graphene is not highly distributed, and it is more tightly present in certain pore area, which size is ranging from 5 micron to 40 micron. So the 0.5% graphene gives a better percentage value in terms of a particular size of the pore. So pore size is more controlled. It's like, I wouldn't say that's very high control, but significantly controlled compared to only collagen and lower concentration of the graphene. And this is overall the analysis what we have done. The another important feature what I want to discuss here is the porosity. Okay, the whenever you develop a gel, you get the pores. Sometimes pores are closed at the end, and some pores are connected to each other and give the continuity of the porosity in the entire system. So if you look at the percentage by the porous porosity, you see the 0.5% graphene has very lowest percentage of closed porosity, okay, compared to EDC and NHS. However, it is not too low, it is not too high as well. But comparatively, if you see, the graphene is further enhancing the open porosity, compared to it. And because of the fibrous nature of the EDC and HS, it does not form a defined boundaries of a porosity. And that's why you see when we talk about the porosity, which this instrument consider pores, if you look about the open porosity, you find a very less percentage of open porosity in EDC and HS, though there's a lot of free space inside. But when you say the percentage of open porosity in 0.5% or 0.1% graphene, you will find a very, very high percentage. This is actually because graphene allows the formation of a clear porous boundaries. Okay, and that machine determines that this is definitely a pore. Okay, and in case of EDSNH, it is like a fibrous nature. The pores are the boundaries of the pores are not defined, and hence it does not consider it as a pore. Okay, further, the connectivity and degree of anisotropy and fractal dimension is all calculated, which further strengthen the property, the porous properties of the graphene incorporated materials. With this, we've confirmed that the material has electric conductivity, it has sufficient porosity, which is required. The next important thing is the mechanical strength. And to start with, we have done the mechanical characterization by various methods. So we did the rheological studies, which talks about the modulus of the material at various angular frequencies. And here we again found the 0.5% graphene is showing a very much better modulus, which means the at various stresses the strain is adjustable and it can go to maximum strain. Okay, so this determines again the material is quite spongy in nature. Okay, it is not very brittle kind of nature. It's very, very spongy nature, and sponginess increases with the porosity and the type of polymer, and it depends on the concentrations as well. So 0.5% comes out very good again compared to the 0.1. 0.1 is also good comparatively, but we have chosen 0.5 as in for future studies. And similar trend what we have found in our universal testing machine test analysis, we have compressed the gel. okay, in UTM. When we compress, you see the flat line, when we do stress versus strain, you see most of the graphs goes like, in a parabolic fashion on like a linear little bit, but here you see it's completely flat. Okay, it means that it can go to very large extensions up to zero point 0.5 at a very minimum stress. Okay, it means it again confirms that material is very spongy. Okay, and the spinal cord at the same time is a very spongy and delicate tissue. Okay, so it will compress very large upon a very small stress, and similar kind of mechanical feature you can see in the material what we have developed. Okay, so overall when we compare the young modulus, zero point 0.5 showing the better young modulus, higher the younger modulus means more sponginess. At the same time, the material is quite tough compared to the only collagen and 0.1% graphene, as a comparison. So with this, we have made a material having a good porosity, which is important for in spinal cord repair and regeneration. We got electroconductivity. Material is electric conductive in nature. We have mechanical strength. Looks like a very spongy material, and so we wanted to check, what all this material, whether addition of graphene, what it is doing, with the collagen. With this, for that we have done the Raman spectroscopic analysis. The rationale behind choosing Raman is, it gives us the some of those helical features of the collagen, which is actually quite not possible with any other method. It's quite sensitive method, and when we check the graphene and amino functionalized graphene, we want to see whether we have compromised the graphene property, and this overlapping band shows that there is no compromise on the basic features of the graphene, which is important for our material. And when we looked at the collagen, which comes at 785 nanometer, so we see that there is some difference in these signals. Okay, and these signals, the collagen, whenever you do Raman analysis, you will see there are two types of amide regions are very prominent in Raman, that is the amide 3 type and amide 1 type. And when we see what is happening here, we again saw that more is the amide 3 and amide 1 type, means it is more promoting towards the helical content of the collagen. So collagen as such is a triple helix. Is a helical material. But when you isolate the collagen and you get the raw this powder collagen and try to mix it again, you actually distort the helical content. Okay, what we observed that the process what we have used for the fabrication of the material, and in the presence of graphene, we see the graphene is trying to cross-link in a better way compared to alone EDC NHS, which means graphene as a cross-linker is actually controlling the collagen helical content. And you see this red line showing the 0.5% graphene is showing significant effect on maintaining, or you can say promoting, the helical content of the collagen. So with this, we confirm that the helical content is increasing. Mean it is further help in the mechanical sponginess of the material, and it also helps in making the material little tougher compared to other. Uh, don't bother this. This is just some of the ratios which are important to understand to make a relative conclusion on this study. And with these ratios, what we have done is to understand the content of the ordered structure in the collagen, and the collagen orderliness, how much it is orderliness, and the degree of cross-linking. And again, with the Raman, you can also check the degree of cross-linking. And again, we have seen the degree of cross-linking is showing much better in case of 0.5% graphene, which further confirm that cross-linking is better. It further indicate that 0.5% is helping the collagen orderliness, and total order structure is much higher in case of 0.5% graphene. You can do all these by just making a ratios of the various intensities in different wavelength. So with this, we have done our most of our material part, and Now we wanted to see in more application. So the first thing is what we wanted to know is whether by, if we add the BMSCs, and we want to see what happens to BMSCs when we give current in the material. What happens to macrophages when we give current. We also wanted to see without current how these material will behave, okay, because we need to know what this material is actually doing based on their mechanical properties, sponginess, or the electrical conductivity. So we have designed this experiment, and for that you see this is setup we have made in house. This is a petri plate, I think, yeah, 6 well plate, and we have added the electrode, and there is a carbon electrode which goes inside in the well, and we fit the material between the electrode, and we keep this, and we control the voltage in this way. And this is a closer look where you can see how we have made this petri plate as our small bioreactor, or system which can give the electric current. So after electric current, we wanted to see a few things, that what is happening with the astrocytes, okay, what is happening with the ATP. Now we have chosen ATP for some another reason in this experiment. The reason is we have seen that ATP is actually energizing the neurons, and there are few reports that the higher, even there are some approaches we have heard from the clinician that ATP injection has been given for the treatment of spinal cord injury. So we want to see whether without any treatment can we have a material in which the stem cells, when stimulated with the current, can show a higher ATP release. And this is what we have found. We found that at 0.5% graphene, when we give the electric current at day 2 and day 3, you see suddenly the ATP concentration is increasing. Okay, and this is something adds on to feature of these materials. And we have checked the expression of CD90 and CD70, which further confirms that the stemness of these materials, these two markers are actually for the stemness. And we see the stemness is actually slightly enhanced when we give the electric stimulus, and at day 5 we found some significant differences. So it is not actually killing the stem cells. The stimulus also, it is not even without stimulus or with stimulant, it is not changing much of the expression. It is comparable, the data is comparable with two-dimensional studies compared to the material. But in 0.5% graphene, when we stimulate the current, it shows a higher expression of both these markers, which confirms that BMSC is having a very good stemness in the whole process. Then, okay, if it does so, then what happens if we try to differentiate these stem cells? We again planned experiment. We grown the cell BMSCs, and then we added the neuronal differentiation media. But at the same time, we are not only entirely dependent on the media, but we also given the current. Okay, so after 7 days, till 7 days, we were there in neuronal differentiation media, but then we started giving current. Okay, and what we have seen that the some of the markers which confirms the neuronal differentiations are MAP2 kinase and beta tubulin and nestin. So the beta tubulin and all is a mature neuronal markers, and some early neuronal markers are there, which further shows that 0.5% is significantly increasing the expression of these markers, which further confirms that in vitro that these materials are promoting the differentiation compared to the stem cell alone. Okay, so which is helpful, which is actually open the open a window where we can use material for the delivery of stem cells as well. All right, so I have told you that inflammation is one of the key things. So we want to see what this material will do when there is inflammation. Okay, so we created an artificial inflammation in vitro culture. We have taken the raw cell. Raw cell, you can induce the raw cell with the lipopolysaccharide. When you treat the lipopolysaccharide with the macrophages, then you will see macrophages turn into inflammation. Okay, so first we confirmed whether the macrophages have inflamed or not in LPS. We have optimized this concentration of 10 microgram per ml, and this is a normal raw cell. So it is a cell line. So even the cell line level, you see the various markers are upregulated, but when we induced it, you see a higher expression of many markers. Okay, so these are the markers which are got upregulated. Okay, and based on that, what we did, that we also collected the supernatant and checked, and after taking the supernet of the inflamed raw cell, the media, and then we expose it to the BMSCs, which are actually growing on the collagen, our cryogels. Okay, so material having a BMSCs were treated with the inflammatory media. Okay, and after adding the inflammatory media, we wanted to see whether BMSCs are having this anti-inflammatory properties along with our material or not. For that, we did the kynurenine level. So kynurenine expression, if it is it's going high, is decreasing, it means the inflammation is working. Okay, and anti-inflammatory property of BMSCs is working, and you see that all these 0.1, 0.5, 0.5, 0.1% of graphene incorporated collagen and EDC and HS collagen 2D are showing a similar result. But after I think it's days are missing, but after I think 4 or 5 days, you see complete suppression in many of these. However, we did not see a significant differences between all these things, but it does show that the behavior is not abrupt, and these are actually showing a similar trend what we can observe in EDC and HS crossing collagen. So probably the BMSC's anti-inflammatory property is not modulated so much in the presence of graphene. We wanted to see macrophages polarization. I told you, because macrophages lives in either in pro-inflammatory or anti-inflammatory, and there is a delicate balance between these two. So if we did cell attachment study, we saw that cells are nicely attached on both the 0.1% and 0.5% graphene. And what we saw is whether the cells are polarized towards the M1 type or M2 type. Okay, so here you see the 0.5% graphene is with CD86 marker. What we have checked, this is for the M1 polarization. It's quite high. Okay, and for that is at day 3. At day 7, it reduces. Okay, and for the M2 polarization, you see that we both the marker what we have taken CD163 and CD206, the 0.5% graphene is actually increased towards the M2 phenotype at day 3, and the level is further increased at day 7 as well. So it confirms that the material is actually pushing the macrophages towards the the M2 and anti-inflammatory phenotype, which is beneficial. And when we compared this data with the EDC and HS, we found that probably graphene is mainly responsible for this polarization and promoting more anti-inflammatory type of macrophages. We confirmed the same with our immunohistochemistry analysis in the cell in our material, and by using CD206 as a cells, you see more of CD206 are present, and very less amount is present here. You see CD86, which is pro-inflammatory kind of marker. You see very diminished expression is there, so very less pro-inflammatory markers are present at cell level. So now, we anyway did with this all cytotoxicity and all. I'm not going to discuss those. They're very standard. So with that in vitro culture studies, we found that macrophages are modulated. We can control the stem cell phenotype. We can differentiate the stem cell in a better way and with a higher efficiency probably. And then we wanted to move ahead and confirm that how this may behave when they are mixture of cells and like a spinal cord. Okay, so for that we tried doing the organotypic spinal cord splice culture. Okay, in the organotypic culture, what we did, we have taken out the spinal cord from the rat. we have sliced that into something like 5 to 10 in height, in thickness, and we you know put it over the scaffold. Okay, this is a conduit like scaffold. We put the sliced culture. You can see the picture here in the, and then we allowed them to attach and put it in media. And what we wanted to see whether the cells are going inside, whether these materials promoting the attachment of the slice and the material, and how the cells are going inside and trying to grow inside the material, what is happening with microglia polarization, what is happening with astrocytes, okay, and what happens when we give current as well. Okay, so we did the study at days 5, day 7, and day 14, and we have did many the genomics kind of study with RT-PCR, protein level, Western blot, and immunohistochemistry. You will see that we started calling it organ on the culture, on organ on gel culture, and first we want to see whether this panel core sticks onto the material or not. And for that we try to remove this. I don't know whether this video will work as well. No, it's not working. What we are trying here, we are trying by using a forceps, we are trying to remove this slice of the spinal cord from the material, but unsuccessful. We were not able to remove it with any shear shaking and any scribbing. So we observe that this attachment area, we have carefully taken this area and dried the sample and looked under the FESAM in the scanning electron microscope, and you see this material has started forming this, forming a bond, just like a cells have started forming attachment with the surface of the material, which it's like a growth cone. This kind of growth cones you will see. It means the growth has initiated, in the in vitro conditions as well. Okay. So we looked whether this is nicely attaching onto that. It is supporting it is loving the material. It is attaching to the material. Adherence is good. Forming as a growth cone. Everything is fine. So let's look into the inflammatory responses. Okay. For that we did the RT-PCR analysis of GFAP, which is a astrocyte marker, a reactive astrocyte marker, and you see in the collagen alone, you see a very high levels of astrocytes, reactive astrocytes. Okay, but in graphene, so for organ on culture experiment and for the in vivo experiment, we have chosen 0.5% graphene. I just want to remind all of you, and all of our studies are conducted on only 0.5 because it looks like a superior material as per our the material properties and the in vitro studies. So in this experiment also we have only chosen the 0.5%. We started calling GR call. So you see the GR call is showing the significant lowering in the reactivity of the astrocytes compared to the EDC-NHS. And it is completely diminished, actually, you can see, even compared to the 2D cell culture. Okay, so this means the reactivity of astrocytes was suppressed by the material alone. And this we have seen the literature, and we found that graphene has shown the capacity of modulating these the expression of the astrocytes. We have checked it at the protein level as well. Okay, we compared both day 7 and day 8, and overall conclusion is that the GR call is actually controlling the exercise reactivity. It was further done to check the vimentin, which is another marker, and the similar observation was made just like GFAP, which further supports that the GR call is actually controlling the exercise reactivity and decreasing the inflammation. Uh, this is immunohistochemistry analysis, which we further compare. So we try to confirm it various means, Western blot, PCR, and again, you can see the GR call is showing a decrease in the expression of vimentin, compared to the only collagen. Okay, and the significant differences were found, and there were some deviations in the immunohistochemistry analysis as well, where the difference were not so significant in GR call, but the overall trend by analyzing more than 10 or 12 images, we found that GR call is actually suppressing or controlling the exercise reactivity. When we looked into the microglia, so for spinal cord, in a spinal cord injury, the higher ratio of M1, M2 microglia is there. So again, we do CD206 and CD86 kind of marker, and you can see that this CD206 and the expression of CD206 and the CD86 is at day 14 of GR call. You see a very nice control over the expression of CD206 and very less expression of the 86. And here you can see that blue color bar is showing that it does increase slight inflammation at up to day 14, but then it keeps 86 under control. Okay, so the overall ratio, if you see the overall density, integrated density of the CD206 in our immunohistochemistry data, you will see the call has showing the higher expression of the CD206, which means it is more like pro-inflammatory, and it is also showing very high anti-inflammatory, but this balance between the call is not good for the repair process. Okay, but GR call show a very tight control over the expressions of both type of markers. One is pro-inflammatory, another is anti-inflammatory. Okay, so we again wanted to check, when we grow the spinal cord, slice on the material, what happens when we, give current. Okay, so after 7 days of culture, when they nicely attach, we try to, look into the beta tubulin gene, which is, is a mature neural marker, and with the current and without current, when we compare, much higher expression was seen at day 14. We constantly given the this treatment of 50 millivolts per millimeter for 5 minutes per day from day 17 to day 14, and at day 14 we found a very higher expression of beta tubulin, which again say that probably current is further promoting the maturation of the neuronal markers. Okay, which is helpful. We did the Western blot analysis of beta tubulin 3, and nu N is another mature neuronal marker. And in both this experiment, both in the current, at some places we did not found a significant difference with current and without current. But for beta tubulin, we found a significant difference in giving a current at day 14. And same result is apparent. You can see within the various groups that GR call with electric current at day 14 showing a very thick band of, beta tubulin, and here if you see there is not a significant difference, but GR call alone is like showing the difference. So some Western blot is faded off a little bit, but overall again that the GR call is actually with current also, it is promoting the neuronal regeneration and some suppression of inflammation. So with this we have further looked at other markers at immunohistochemistry, and the trend is similar what we have discussed in the previous slide. So I'll skip this. So why this is happening? I mean, there were several questions going on. One, current is what we have seen. We wanted to see what happens to the mechanical signals which we are giving through, because material has this sponginess, how the material cells are behaving onto that. Okay, so for that we did the spinal cord. When the spinal cord slice was cultured onto the material, we actually tried to see the mechanosensitivity analysis. Okay, again we have chosen 3 very important genes: paxillin expression, the focal adhesion kinase, and vinculin. So these all 3 genes are named as mechanosensitive gene, and they regulate the expression of the cells. So again, both particularly GR call is showing a significant increase of paxillin and focal adhesion kinase. Even collagen is also not so far behind, and for focal adhesion kinase is also showing a better adhesion kinase expression in focal adhesion kinase expression as well. In case of Vimentin, we did not see a much expression of the GR call at 14 day, but collagen maintained the expression more or less just like at day 7. So here with this, that okay, this combination of collagen and graphene together is actually modulating the paxillin expression and focal adhesion expression, and which actually can give us more detail in our future experiments. We haven't looked at now, but we'll further interested in how these mechanosensitive genes are actually controlling the various expressions of the macrophages or probably astrocytes and probably stem cells as well. So this is something very interesting we have found. Which is useful to understand the material interaction with the cells and tissues. We further then with all these studies, we finally decided to do the in vivo analysis to check whether the material alone can do the immunomodulatory function and can help in the neuronal regeneration. For that, we have selected female SD rats with a weight of around 280 to 320 gram. We do the surgery procedure to make a hemisection model. Okay, so in the hemisection model, you just cut a piece of it, not the complete section of it. Okay, with this we have implanted. This is our animal group. We have taken all the post-operative care for the animal, and we have implanted the material, and then we seal the parts, and after 28 days we have isolated the spinal cord, and you can see the material is nicely embedded, made a nice junction, both in collagen and GR call. Okay, collagen we kept as always as a control, and injuries, of course, you see unrepaired parts are there. So with this, we so to confirm that we have done the functional locomotor behavior analysis, and I have done this study with my collaborator, Dr. Hemant. So you see, this is a very nice video, and this clears everything. Okay, so this is an injured animal. Okay, this is a sham. You see, even after 28 days, this leg, you see, it is not straightening up. It is just like a limbing, and it is not able to walk. When we added the collagen alone as a material implant, then you see it started walking a little bit, but still the right hind leg is not fully recovered, and it is still slightly paralyzed and not so useful. Okay, but you see the GR call. When we implanted the GR call material, you see the rat is moving, is everywhere. Its paws are nicely placed onto the surface. This is all observation. I'm sorry if the video is not so clear, but this is our clear observation that you see both these legs are nicely flattened onto the ground and helping in walking and all. And based on that, we have done some functional analysis, and so the very standard BBB scoring. So within BBB scoring, we got a very exciting result that the GR call, the blue line, if you see right after day 7 to day 14, it just shows the uptrend in showing the locomotor behavior of the animals. Okay, and just in 14 days, this is a highly significant time point where such a jump in the locomotor function was found and remain up compared to the collagen and sham alone. So we looked into the structure. This is very exciting. So we found that for locomotory recovery, up to this point, when we achieve like more than 10-12 score, how many groups have scored that much? So we see like some material we have identified recently published papers. We found they show the time taken for some locomotory recovery is 12 weeks, 6 weeks, and 8 weeks. This is what, and we have achieved it within 14 days, and this is something really exciting for us. And in our studies, within less than 2 weeks, it is achieving the locomotor function, and very good locomotor function at 28 days. I mean, after 4 weeks is very good. Okay, they are showing the rat is running here and there. Further behavioral analysis, so there was not a significant difference in the grip strength. So grip strength is fairly same in all. So looks like even in the injury animal, the grip strength is not so compromised, and that's why there is no significant difference between any of the group. When we see the power withdrawal capacity, that is on the hot plate analysis, we see just like sham. Sham means a healthy animal. So a GR call is just like a sham, compared to injury and the collagen implanted. So again, GR call achieved a milestone here to control the neuronal functions. On a hot plate cold, the rat withdraw its legs upon the temperature. So this is very exciting, and then we further compromise the animals, and looked into the immunohistochemistry way to see whether they have repaired the injured spinal cord or not. And here you can see this is an injury group. You see we have marked the injury area with this yellow dotted line. This is day 28. We have compromised animal. You see there is no neuroinflammatory marker expression, no beta tubulin expression here. Okay, but in collagen you see this is a collagen material is there, very low beta tubulin marker, very low neuroinflammation. Though it made a connection, it tried to make a connection, but overall, when we see the GR call, GR call shows a very high expression of beta tubulin 3 and neuroinflammation, and trying to make a full junction in the spinal cord. Okay, so we have done the intensity analysis, and again we found a GR call showing the significant increase in the mature neuronal markers, so that were beta tubulin and the NF200. What about the microglia? So we looked the microglia, and you see the 206 and 86 is the marker what we have chosen. So you see that 206 is quite high in the collagen, and there is some expression of CD86. In the lesion area, what you see is the CD86 is very high. Okay, in GR call, you see both are nicely controlled. Very less expression of 206, which further confirm is just like a not pro-inflammatory. So collagen is showing some pro-inflammatory kind of expression of CD206, but CD86 expression is like higher than the collagen. Okay, if it fully grows, then maybe for a prolonged period of time, if you keep, it may increase the CD86 expression as well. So this shows that it is still controlling the inflammation in vivo condition. What about the another very important inflammatory cell, which is astrocytes? And we were happy to see that the astrocytes reactivity is significantly lower in the GR call, in GR call compared to the injury and the collagen alone. So overall, the developed material, the graphene collagen material, is actually helping in suppression of neuroinflammation and promoting the growth of the spinal cord cells. So again, it is like more of an observation thing, like why it is we are putting things and trying to see, and this is happening. So we want to see why this is happening, and we try to connect it with the very important pathway called STAT3, and it is seen that STAT3 expression is actually help whether when the STAT3 expression is higher in neuronal and spinal cord area, you will find that it will have a better neuronal regeneration, as well as more anti-inflammatory kind of pathway is modulated. Okay, so in the GR call, you see the STAT3. Sorry, I just say the higher expression. I mean to say lower expression of STAT3. So when you compare the injury area, very high expression of STAT3, which means it is highly inflammatory and area. And when you say collagen, it able to control some of the inflammation. But when we use GR call, it further lower down the STAT3 expression. And when the STAT3 expression is going down, it can actually control the inflammation by controlling these statocytes and probably the macrophage polarization as well. So we try to link it with the STAT3 expression, whatever observation we have seen, and we are currently trying to understand more mechanistic view of why this is happening through the next generation sequencing. The data is not available yet. We are still analyzing it, but that will give us a clear picture that how this inflammation is controlled by just giving material as an implant. Okay. This is very important. Again, like, when we want to say whether the spinal cord is regenerated or not, we must do the myelination estimation, okay, and how much is the lesion capacity. So we did the routine HNE analysis, and when we see the collagen GR call, you see not nice tissue integration with the thing. Here you see only a part of the material is integrated and that's it broken off. But here, very nice integration you can see, and inside you this cells are coming. So in the entire material you see the cells are proliferating. Okay. Uh, Here you see the LEB staining. This is a sham which is showing a good myelination formation. Material is it is collagen. This is injury group, and here you see the GR call, which is showing a nice myelination sheet are forming. Okay. So sorry, this is the injury group. This is just a an unrepaired area. All right. So the myelination is also possible to be like we can enhance the myelination in the injury area by giving a material, and probably this myelination can be enhanced when we deliver the stem cells into it. We further try to look into the ECM deposition. For that we have chosen two very prominent marker, fibronectin and laminin, which is actually important to provide it is a home ECM in the injured area. And again, when we compare the GR call, when we see the GR call, there's a good expression of laminin and the fibronectin is found, which further support that material is so porous. It is providing a very nice framework where the cells can grow, matrix can be secreted, and it's trying to replace the material with the new spinal cord. Okay, so ECM matrix remodeling is also observed by providing these materials, and we have also seen the both the broken ends are trying to connect through the material, and for that we have also seen the orientation of the cell. When two cells trying to connect each other, its orientation is quite different, and the growth is not random. Okay, so we have taken the spinal cord, so there is a plugin is available with image J. Okay, and we try to see based on the DAPI, we can see that whether the nuclei is oriented in particular direction or not. When we see the sham spinal cord, we see it is nicely, it's single peak, a sharp single peak is demonstrate that this is in good alignment, and the similar alignment was found in GR call. However, call is also showing the alignment, but the level of alignment is not as significant as what we get in the GR call. So this kind of image-based analysis further supports our data and our claim that GR call probably provide a electroconductive of material, electroconductive mechanical spongy material for the spinal cord repair and regeneration. Okay, so we're still doing many other experiments to confirm whether GR call can be used as a implant material for spinal cord regeneration. We have also conducted the BMSC's delivery using. I'm not showing that data here. It will go over time. So BMSC's also shows that in some experiment it is showing the better efficacy, but in some experiment it is as equivalent as GR call alone. So we are further analyzing the data. We have also have NGS data. So altogether we will be probably having a better mechanistic view that how this whole repair process and inflammation control is going on. Okay, so this is the overall summary on our work. So the challenge was the scar formation, which is because of the reactive exercise, the phenotype of macroglia, which further does not allow the repair process, and the hindered regeneration, which is a like capacity of the neuron. But with our material, what we are able to achieve, we are able to achieve the neuronal development because we can decrease the exercise reactivity. We can have more anti-inflammatory type of microglia polarization. The ratio is shifting towards the anti-inflammatory, and hence we can get the accelerated neuronal regeneration because of the interconnected pores. Okay, so with this overall summary, I would like to finish my talk, and I would like to acknowledge mainly my PhD student, Dr. Gopal. He's just finished his PhD and moved to the Florida for the postdoc. There are many master students. I cannot list them out here. And I would like to thank my collaborator for my all in vivo based studies, Dr. Hemant. He's in the same department as a assistant professor in the Department of Pharmacology and Toxicology at NIPER Ahmedabad. And I am thankful to all the generous support I get from the NIPER Ahmedabad. I finished the DST SERB Early Career Research Grant. I'm a DVD Ramanlinga Swamy Fellow. So that grant I want to thank for. And recently on this particular work, I got the core research grant. And for all those imagings and many other testings what we have conducted in the center and instrumentation facility at NIPER Ahmedabad. Thank you very much. I would can have few questions if somebody have it.
Hello.
Yes. Hello.
Thank you, sir, for your wonderful talk. And I would like to thank you for on behalf of as a convener and organizing team and on behalf of the Department of Biotechnology and Medical Engineering Department at NIT Rourkela. So you gave a clear and clear cut, very prominent research information about how uh, the spinal cord injury you can the recover and other things. Okay, so it was really a great lecture. So I would like to with this lecture I would like to the open up the platform for if you have any questions, please feel free to ask. If you are willing to ask any doubts, do you have any do you need any clarification about the work and other things, please feel free to ask any questions that you have. I think, Prasun sir, you are asking some question, but we are not getting it.
Yeah, I was asking something. Hello.
Hello, Prasun. Yes.
Yeah, Akshay, how are you?
I'm
Yeah,
good.
it was indeed a very good lecture. At least I have witnessed some part of it when I was there. Uh, Actually, I have one small question. So you have used the graphene as a material,
Yes.
right? So the role of the graphene was to enhance the mechanical as well as the electric conductivity, right? Or
Yes.
it has to offer something else as well.
So that was the basic agenda, that we thought that graphene will be a good material because we want to increase the electric conductivity. That was the prime aim. We thought, because didn't wanted
Okay.
to choose. We compared the other polymers, but we found graphene is like a small and give a better properties, and also it can give a better sponginess compared to other polymers. So we incorporated with the collagen. But when we
Okay.
started doing the cell culture studies, we found the graphene can also influence the cellular response. Okay, then we looked the literature and found there are recent reports where graphene alone as in the form of particles have shown to modulate the inflammation as well, some ROS activities also. So that was a
Okay.
plus end which we get to know later on.
So it's a composite of the collagen and the graphene, or it's a
It's a
graphene
cross-linked.
grafted or something like that. What
cross
was the
cross-linked.
crosslink? Okay, okay.
So there's a chemical cross-linking between carboxyl graphene. So we have modified the graphene to carboxyl graphene, and we have amino
Okay.
group. No, sorry. Yeah, amino graphene, amino group, and carboxyl of the collagen. We cross-link with the
Oh,
carbonamide.
crosslink. Okay, okay. Okay, okay. Thank you, Akshay.
Thank you, person.
Anybody else would like to ask any question?
I think they all slept.
So actually, it was indeed a very good lecture, but more technical rigor. And I think in the second half, something very technical people are not used to. I don't know. But the last part I liked very much, when the mouse was able to move
Yes.
and crawl, and that was indeed at least it gives the pleasure. Okay, every
Yeah.
material, every mechanism we have done, but it is working somewhere. Yeah. Okay.
Hello, sir.
Yeah,
I
somebody
have
is
one
there.
question, sir.
Sure, go ahead.
Hello. Sir, can we use this material for other inflammatory diseases?
See, I mean, it is not a very generic material. We have tested in the spinal cord because there is a clear mandate of controlling the inflammation. Rationale is there. But when we talk about the other inflammation, it is also depend whether those inflammations are because of astrocytes or macrophages. Then probably we can try exploring that. But it's not very general material that you use everywhere. Because in the starting also we looked into the problem, spinal cord problem, and then we try to design the material. So it is kind of a inspired kind of a material which you have developed, but it can be explored. It's like a testing we can do, but I cannot say that it will be fine with any other disease.
Thank you, sir.
Okay, any other questions? Okay, so Akshay, I think
Yeah,
there is no more questions. But if anyone is having any questions, any doubt, you can message the question. We will forward it to Professor Akshay, and he would be happy to address your queries.
sure.
And thank you, Akshay, for giving such a insightful talk about the. And we will probably see some of this material going into the clinical trials. I am at least hoping for the same.
True.
And
For large animals,
uh,
so it can help me.
yeah, definitely large animals, and then clinical trials. Yeah, it's a long way, but yeah, means journey has started.
True.
Yeah.
Thank you very much. Thank you, Dr.
Okay,
Manoj.
Akshay.
Yeah,
On the behalf of the organizing team and the committee, I thank you very much for giving a lecture and joining with us.
yeah. Thank you. I want to thank you.
Thank you.
Okay, bye.